Hello, this is Dr. Sandeep Sarkar, Assistant Professor from Holy Cross College, Agartala. In today's class, I am going to describe about the regulation of gene expression and the Lac Operon. Is the regulation of gene expression and the Lac Operon. What are genes? Genes are those part of a DNA which are expressed in the form of some proteins, and the genes are the functional part of a DNA. That genes are expressed through either transcription or the translation, but which will decide when, how and where these genes will be expressed. Actually, this expression of genes can be regulated in many ways depending on the cell organization and the need of the organization, the environment of the cell, the environment of that organism, which decides when, how and where and which gene will be expressed. A prokaryote is totally controlled by its environmental changes. Its surrounding environment may be the outer side or the inner side of its body. The central dogma of a gene expression is from DNA to RNA by the process of replication, from the mRNA to the protein by the process of from the DNA to the mRNA by the process of transcription, and from the mRNA to the protein by the process of translation, and then we can say that the gene is fully expressed. The two well-studied mechanisms of transcriptional control of gene expression, one is the Operon system, another one is the cascade of gene expression. So what is the Operon system? It is a system where the genes involved in a metabolic pathway are regrouped into a gene cluster which controlled by common regulatory sequences and the proteins. The expression of these genes are then rapid and are synchronized. This is the Operon model, which was developed by two scientists, Jacob and the Monod, in 1961. The another mechanism is the cascade of gene expression. What is that one? This is the model where under some environmental conditions, expression of a first set of genes can be switched on, and one or more of the products of this first gene set will switch on a second one, and this event could be repeated many times to mobilize wider gene sets to achieve a special metabolic pathway. So that is the two types of transcriptional level of control of gene expression. One is the Operon level and another one is the cascade of gene expression. The gene expression can be positively or can be negatively controlled. So what is the positive control? The positive control is that system where the binding of activator protein triggers the transcription, whereas in negative control system, the binding of a repressor protein inhibits the transcription. What is the difference? I repeat again. In case of positive control, binding of the activator protein triggers the transcription. In case of negative control, the binding of a repressor protein inhibits the transcription. That is the difference between the two types of control. So now let us enter into the Operon concept, the Lac Operon. What is the Operon concept? What is the Operon? An Operon is a group of genes that are transcribed at the same time. An Operon is a group of genes. There are not a single genes. More than one genes are involved and that are transcribed at the same time. They are usually control an important biochemical process, and which is found only in case of the prokaryotes. It is a negative control where binding of repressor protein inhibits the transcription. Where the binding of a repressor protein causes the inhibition of the transcription, that one is a negative control. Which one? Actually, I am talking about the Lac Operon. So let us see what is the Lac Operon exactly is. This is the Lac Operon. The organization of Lac Operon DNA structure. The Lac Operon DNA actually consisting of two broad groups. One is the structural genes, another one is the regulatory genes, or the structural loci and the regulatory loci. The structural loci is having 3 genes. They are named as gene Z, gene Y, and the gene A. The gene Z code for beta galactosidase, gene Y codes for beta galactoside permease, and last one, that is gene A, codes for beta galactoside transacetylase. These are the 3 genes which codes for 3 enzymes which are present in the structural loci. What about the regulatory loci? The regulatory loci consist of a promoter region for the Lac Operon structural genes, then a operator region for the Lac Operon structural genes. Along with this, the regulatory loci also contains a I gene and the promoter for that regulatory I gene. The I gene is actually the regulatory gene. It is present in the regulatory loci plus the promoter region for that I gene. So this is the structure of a Lac Operon DNA. The Lac Operon consists of 3 genes, each involved in the processing of the sugar lactose. Since we know that lactose is the sugar, it's not a monosaccharide, it's a disaccharide lactose. And for the processing of lactose, actually what is the condition is that when the prokaryotes, mainly the bacteria, they require either glucose or the lactose for their energy purposes. So when lactose is present in the medium, since it is the disaccharide, so that prokaryote, that bacteria needs some of the enzymes to degrade the disaccharides into monosaccharide, and then that lactose should be transported throughout the cell, and then it will give us the energy to that prokaryote. So for that it requires 3 enzymes, and for each of the enzyme there is a particular genes. So the Lac Operon, which consists of 3 genes and each involves in the processing of the sugar lactose. First one is the beta galactosidase, which codes by Lac Z gene. What is its function? It cuts the lactose into galactose and the glucose. Second one, which is transcribed from the Lac Y gene, the enzyme is galactose permease, which transports lactose into the cell. The third, that is Lac A gene, codes for galactoside transacetylase, whose function is not fully clear till now. The E. coli can use their either glucose or the lactose. So glucose is a monosaccharide, but since the lactose is a disaccharide, so for the use of lactose as a energy source, the prokaryotes, say for example the E. coli or the bacteria, needs these 3 enzymes, therefore these 3 genes, and that is the importance of Lac Operon in case of this organism. This Lac Operon also having inducer and a repressor. What is the repressor? The repressor is an allosteric protein which is synthesized from the I gene, which is a part of the regulatory loci, I gene. So from that I gene, the repressor is synthesized. That repressor is a allosteric protein which is 36 kilodalton in molecular weight. What is its function? This repressor protein binds to the operator site. Which operator site? The operator site which is responsible for the structural gene. So this repressor synthesized from the I gene binds to the operator site as a tetramer. Binding to operator sequence is extremely tight and specific, and this causes the transcriptional repression. So that is the repressor. Another one is the inducer. So what is the inducer? Inducer is an alternative form of lactose. Inducer or the allolactose, which is an alternative form of lactose that binds to the Lac repressor and inactivate it. Inducer is actually an allolactose, which is a form of lactose. The allolactose is a lactose related compound that is produced by the basal level of beta galactosidase. So now this inducer binds to the Lac repressor and thereby inactivates the Lac repressor. So what is the net result? The net result is that when the inducer binds with the Lac repressor, which causes the Lac repressor to become inactivates. Now this Lac repressor is not able to bind with the operator site for the structural genes, thereby the operator site is remain open, and there the DNA polymerase can bind and it can transcribe the gene. Therefore, when the bacteria needs, it will synthesize 3 of the enzymes. So that is the thing. Okay, let me describe again. See, look at the Lac Operon DNA gene. Look at the Lac Operon DNA. In the structural loci, there are 3 genes for the synthesis of 3 enzymes. In the regulatory loci, there is operator and the promoter site. So from the I gene, the repressor is synthesized. Look at this structure. From the I gene, the repressor is synthesized regularly and it will bind to the operator site. So when the repressor will bind to the operator site, the DNA polymerase cannot bind, the RNA polymerase cannot bind with the promoter site, and it is not able to transcribe the structural gene ZYA. But another allolactose, which is the inducer, when the inducer binds with the repressor, it inactivates the repressor. Let me describe with example. These are the example. First condition, when lactose is present. So what will be the thing? When the lactose is present in the medium, the food or the energy source for the prokaryote. Now the prokaryote needs to break down that lactose disaccharide and utilize it as a fuel for generation of its energy. That means for the use of the lactose, it triggers 3 enzymes: beta-galactoside, permease, and the trans-acetylase. So for the formation, for the generation of these 3 enzymes, that Lac Operon DNA, that Lac Operon genes should be transcribed. Now the process is that from the Lac I gene, the repressor is always transcribed. When the repressor is bind with the operator region, the RNA polymerase is not able to bind at the promoter region and the genes are not transcribed. That is the normal thing. Now we are having a condition when lactose is present. So when the lactose is present, that means the inducer is also present, because if the lactose is present in the medium, the inducer, the allolactose, is actually a form of lactose which is formed due to the activity of the basal level of beta-galactosidase. So when the lactose is present, the allolactose inducer is present. Now what is the role of the inducer? The inducer binds with the repressor protein and it causes the repressor protein to become inactive. Look at the repressor protein, the red color, the red structure is the repressor protein, and the inducer allolactose is the yellow, not the yellow, the green dot. So when the repressor protein is inactivated by the green dot, the inducer, it causes the inactivation of the repressor protein. Now the repressor is not able to bind at the operator region. If it will not bind at the operator region, the RNA polymerase is binding to the promoter region, and now it causes the translation of the 3 structural genes, and we are getting that organism will get the 3 enzymes for the degradation of the lactose and the utilization of lactose. So simple thing, when the lactose is present, the inducer allolactose is present. This inducer will bind with the repressor. It causes the inactivation of the repressor. If the repressors are inactivated, RNA polymerase can transcribe the Lac Operon structural genes and the 3 enzymes will be synthesized. Second condition, when lactose is absent. So when the lactose is absent, the inducer is absent. So if the inducer is absent, there is no one which can inactivate the repressor protein. So now the repressor protein is able to bind with the operator site and it blocks the binding of the it hampers the binding of the RNA polymerase to the promoter site. Since the RNA polymerase is not able to bind with the promoter site, the structural genes are not synthesized. And actually the need of the demand of the time is that since there is no lactose, there is no need of synthesis of the 3 enzymes. So when lactose is absent, 3 enzymes are not synthesized. How they are not synthesized? The inducer is not there and the repressor is active. That active repressor binds to the operator. Therefore, RNA polymerase is not able to bind and the genes are not synthesized. Third condition, when both glucose and lactose are present, what will be the situation? When both glucose and lactose are present, the RNA polymerase can sit on the promoter site of the Lac Operon, but it is unstable and it keeps failing off. Why it is happening? The logic behind this can be explained as since the glucose, which is a monosaccharide, is present in the medium. So because of presence of monosaccharide in the medium, there is no need why to go for the degradation or why to go for the cleavage of the disaccharide lactose and to convert that lactose into glucose and then utilizing energy from the glucose. Since we already having the glucose, since that organism that prokaryotes is already having glucose in its medium, so it can freely use that glucose. Since the simple source is glucose is directly available in the medium, so it will go for the glucose. It will not go for the conversion or degradation of the lactose into glucose and then conversion of the glucose. Since the glucose is there, no need for that enzymes, no need for the Lac Operon to become active and produces 3 enzymes for the degradation or utilization of the lactose. So the condition when both glucose and lactose are present, the RNA polymerase can sit on the promoter site for the structural genes, but it is unstable and it keeps failing off. So there is no synthesis of the ZYA genes, no synthesis of the no production of the 3 enzymes and no utilization of the lactose as glucose is already present there. Okay, that is all about the very simple way I have tried to describe the Lac Operon. If you have any question further, please ask or please write in the comment box. I will definitely answer and try to solve your queries. Okay, thank you for today. Goodbye.